Landon's a PhD student in Dr. Chen's lab, so he works with a lot of mechanobiology work. He's coming here today to talk to us about his update on his work. You're a fourth year student now, so he's made progress. Was here last year, gave us like an update on your specific A1, so we're just excited to see where you're at now. Please give Landon your full attention.
All right, well, we'll just jump right into things, try not to take too long on everything. But as Nick said, I work with Dr. Chen in the department, and we do a lot of research on GBM and analyzing the mechanical aspects of cancer biology. And so for those who are maybe a little less familiar with GBM, what I'm referring to is glioblastoma. So this is going to be a primary brain tumor, and cancer in a whole accounts for about 1 in 6 deaths worldwide. It's characterized by this uncontrollable growth and proliferation of these cells that eventually spread to either other regions of the host tissue they're in or entirely new sets of tissue. GBM is an especially deadly type of cancer. While not as prominent as, say, maybe lung or breast, it still has a remarkably dismal prognosis with a median life expectancy of only around 15 months, 5-year survival often being under 5% for those who do have it. And so, in part, leading to this high rate of mortality are 2 primary factors that I'll talk about that have helped to shape the direction of my dissertation. And those are going to be that GBM is a highly invasive type of cancer, and that it is a highly immunosuppressive type of cancer. And so when I talk about invasion, that's going to be similar to metastasis, which you'll see in other types of cancer. Now GBM almost never exits the brain environment. It's extremely rare. So when I talk about GBM invading, I'm just referring to it spreading throughout the brain tissue. And what you'll see here is just a schematic of what this would look like here, where you'll have the primary tumor and then these disseminating, often what we call tumor initiating cells that spread out to form secondary tumors. And so you can imagine, when we know that cancer therapies include resection, chemotherapy, and radiotherapy, and these are often used very wide scale in the tissue systems to eliminate all the tumors, that's a very challenging application to apply now to this brain environment, because this controls everything that you can do as a biological being. So we have to try and balance the need to remove as many cancerous cells as we can, but also limit the potential harm that we cause. And so in this figure right here, you can see how this tumor, it had the standard therapies applied to it that I mentioned earlier, and you can see within a few weeks after taking the image, it was completely gone. But then just a few weeks later, you can see a new tumor had formed on the other hemisphere of the brain. So again, we can't just keep going and cutting out swaths of brain tissue without risking some permanent damage here. And so that has spurred the need to develop these new therapies that are not quite as damaging to the brain, but are still just as, if not more effective than the traditional methods. And so the other aspect that contributes to GBM being difficult to treat is that it is a highly immunosuppressive and immunoevasive type of cancer. And when I refer to immunoevasion, I'm referring to how pathogens, or in this case, the cancer cells, are just able to evade the host immune systems, whether it be the soluble agents they secrete or the immune cells themselves. Classically, you'll see cancers are divided into one of 2 categories, depending on their response to immune cells. They'll either be known as hot tumors, indicating that they are very responsive, or cold tumors, which is what GBM is. And so in the diagram, you can see how we have 2 of the more common types of tumor eliminating cells, the CD8 T cells and the natural killer cells. In cold tumors, they are unable to really penetrate into the tumor and eliminate those kind of proliferating cells that contribute back to other tumor formation. And so knowing that these 2 factors are contributing to GBM's difficulty to treat, I wanted to explore, are there any factors that are extrinsic to the tumor that are contributing to these behaviors. And so, as I mentioned earlier, I work with the Chen lab. We do mechanobiology. We're interested in the mechanical aspects of the environment. And this is going to be in reference to the tumor microenvironment, which is classically divided into two compartments. The biomolecular is going to be what most people are probably familiar with. These are going to be what your biologists are working with: cytokines, metabolites, anything soluble that you can just add to media and try and make your cells undergo some kind of transition. Whereas our lab and my focus is on the biophysical. So I'm looking at a multitude of factors, including the ECM composition and structure of this environment, the proteins that are available to do binding, how the tumor compresses or expands on the cells, how the different fluid pressures contribute to these aberrant behaviors. All of these factors, while they're beginning to be studied, are not very well understood, not nearly as much as the biomolecular side of it. And so this is really because this biophysical aspects only emerged really in mainstream in about the last 20 years or so. But even in that short amount of time, as I said earlier, we've begun to understand some of these factors. We see that tissue density and the ultrastructure do play a role in driving some of these cell behaviors here and regulating the processes. And this is a little intuitive to understand because you can see how in this schematic here, you have a normal brain tissue environment versus a GBM environment. And you can see things are much more densely packed in there. You've got the cells trying to spread out and form the tumor. It's not hard to understand that there could be some changes going on in here. And we see that when you culture these cells on different substrates of increasing stiffness, you see that their cell speeds often increase. And I'll talk about this a little bit more in my AIM-1. We also see that within the tumor itself, there's a high degree of heterogeneity in terms of the mechanical properties, where you can see in these human patient samples that we processed in our lab, we have what we call the tumor core, and we have that invading rim of the tumor. And you can see how there are these wide spreads and significant differences between the two regions. And so all of this has helped to guide me in coming up with this dissertation that is focused on examining this invasion and immunosuppression capacity of the tumor, but from this biophysical lens. And so my dissertation is really divided up into two main categories, with the first one focusing on the tissue ultrastructure side and seeing how these cells are able to invade throughout the system. The latter portion of my presentation is going to focus more on the spatially heterogeneous stiffness regions, which are really focused more on that immunological kind of side of there. And so firstly, I'll talk about AIM-1, which is the work that I began when I first arrived here and am nearing completion on. So this is really going to be focused on achieving the completion of two goals, where I want to identify these biophysical changes in GBM that accompany this process known as proneural to mesenchymal transitions. And then I want to see to what degree are these biophysical changes occurring and can I potentially reverse them. And so, as I mentioned, GBM is a very invasive type of cancer. And accepting that as a fact is fine, but it's a little bit paradoxical when we consider the environment that GBM is in. As I mentioned, the brain is in charge of regulating everything in your body. And so there are vast networks of neural cells and connections between them that are needed. But the problem is when you create this network, you get a very dense system. Pores in this brain tissue are often on the nanometer scale. And when we consider that cells often average on the micron level, that's a 1,000 factor difference that these cells are somehow squeezing through and forming these tumors. And so this further kind of helps demonstrate some of the difficulty with applying these treatments. Chemotherapies are unable to adequately go through these pores, apply the agents to the cells, and so forth. But in having this dense network, I wanted to see, why does GBM navigate it so easily, and are there specific types that are able to do so? And so GBM has been historically classified into 4 main subtypes, although 3 is becoming a little bit more prominent as 2 of them are coming together. But the 2 I'm going to focus on are still in their original forms from those 4 groups, proneural and mesenchymal. And if you want to think about these as just being opposite ends of one spectrum, you can. Proneural is going to be your more easily treated type of GBM, often having a better patient survival rate, whereas mesenchymal is going to be on the opposite end. And so GBM does undergo a plastic transition where it can go from proneural to mesenchymal, potentially even back, maintaining characteristics of both. We see that mesenchymal GBM is one of the subtypes that has the worst prognosis among them. And so I'm going to focus on analyzing what I call these proneural to mesenchymal transitions. If you work with cancer, you're probably familiar with EMT. So that's the epithelial to mesenchymal transitions. This is essentially just the GBM's variation of that, and it occurs naturally as disease progresses. Notably, it's also inducible via TGF-beta1. So this is commonly secreted in disease progression, helps contribute to that tumor evolution in there. And it's going to be a cytokine that I use to help induce these transitions myself. And so taking this all together, my general hypothesis for specific AIM-1 is that if I can induce PMT and just mimic this disease progression, I would expect to see in the GBM a softening of their mechanical properties that allow them to bypass these pores they otherwise wouldn't be able to. And so in doing so, they can spread, form secondary tumors, and just decrease patient quality and survival. I use a host of classic biological assays, and then I eventually leverage some bioengineering tools, including AFM and these microchannel devices that we make in our lab. Just to talk about the microchannels real quick, you can see how they are labeled as 5, 10, 20, and 50, just referring to the width of the channels. 50 microns is going to be more reminiscent of a classical free motility type of assay, whereas 5 is going to represent a much more confined environment for the cells. And so the first aspect of my dissertation was looking into, can I induce these PMT results in my cells? And so when applying TGF-beta to 2 human GBM cell lines that we have, U87 being a already more mesenchymal-like cell, and U251 being a more proneural-like cell, I showed that when you look at the SMAD2 pathway, you get an increase in activity for phosphorylation. All this signals is that the canonical signaling pathway and receptor binding known for TGF-beta was being active in there. Furthermore, I saw that STAT3 also had an increase in phosphorylation events. And so STAT3 is a non-canonical pathway for TGF-beta signaling, so it just confirms some of the TGF-beta binding occurring, but it's also kind of the initiating pathway for these mesenchymal transitions to occur. And so once again, in both cell lines, you see increase in activity, which was finally concluded with this look into ZEB1, a full-on mesenchymal transcription factor produced by mesenchymal-like cells. So taken together, these demonstrate that I could induce PMT in my cells. So the next step was to see what are the changes that correspond with these PMT. And so what you'll see in these figures are that I looked at 2 proteins, lamin AC, which is going to be your structural protein in the nuclear region of the cell, and then the F-actin and G-actin ratios, which are going to be those structural proteins everywhere else in the cell. You'll see that in both cell lines, there are general trends of decreasing expression of both proteins to varying levels of significance, as well as some alterations in the organizations of the cells and their proteins. You'll notice that the F-actin strands become much less pronounced after adding TGF-beta. You see the nuclear regions of the cells often undergo an increasing amount of irregularity after being exposed to TGF-beta. This correlated with a decrease in circularity, and when you look a little bit closer, you'll see the formation of these pore-like structures in the nuclear envelope here in this lamin kind of shield across. And so when you take this together, intuitively it all makes sense. Lamin expression is overall going down. All the organization is being altered. You're getting these pores forming. You're eventually having a less stable nuclear region, thus the nucleus becomes a little bit less circular. Overall, that could indicate that you are getting something that has a less defined shape on it, which would be advantageous for navigating these small pores. And so after seeing these protein level changes, I wanted to examine, are these correlating with now these reductions that I hypothesized in the mechanical profiles. And so I use AFM, which is just a type of microscopy that uses a probe to indent into a surface. By using Hooke's law and the equation for Young's modulus, you can back calculate forces, stresses, strains, etc., onto the cells. And so in both cell lines, you'll see that in the cytoskeleton, which will typically be the darker colors, and the nucleus, which are the lighter, for both cell lines, they're a little bit more spread out. But once I add TGF-beta, those histograms become much more condensed into these softer regions, which is further illustrated in part C, where you'll see in both cell lines, going from non-treated to the TGF-beta treated cells, there's an average of around a 40 to 50% decrease in the Young's modulus, indicating that these cells are indeed becoming significantly softer. And so finally, I'm trying to see, does this correspond to that gain of functionality that they can bypass these confined microenvironments? And so I've included 2 videos here just to demonstrate what these channels look like. And so on the left, you'll see a 50 micron wide set of channels. And you can see these cells are otherwise not having any difficulty traveling through them. Whereas when you look at the 5 micron wide channels, you see there's a high degree of deformation that is needed in order to get into these channels here. And so what's interesting is that when we look at these, the U251, or that more proneural type cell to start with, we see significant increases in cell speed across all the channels, indicating that they are adopting that more mesenchymal-like behavior here. U87s, we see the effects are a little bit less pronounced. We see sometimes they favor a little bit faster, sometimes they favor a little bit slower. And so I'm working through some iterations of this to try and get a more representative image of it. But unfortunately, there was a delay due to power outages on campus and loss of cells. So I'm working through that right now. But overall, at least with the U251s, the ones that would progress through this process more fully, we see there is a significant increase here. And so after validating each part of my hypothesis and seeing that they are becoming softer, they are becoming more invasive, I wanted to know, could I directly modulate this behavior? And so I took this stiffening agent, RO3306, and it's classified as a CDK1 inhibitor, but it also has some implications in stiffening cells through a lamin network. And so with this drug being used in other cancers and showing that it can modulate their stiffness, I wanted to apply it to GBM as it hadn't been done before. Of course, I had to work through several optimization steps to get the concentration correct, to get the exposure time correct, because as I said, it's a CDK1 inhibitor that is just deadly to the cells in long enough time. What I've included here are the Western blots scans that I took, looking at P-lamin and lamin AC. As I mentioned, it works by a lamin. There's an inverse relationship. When you have more mature lamin that would give you that structural soundness in the nucleus, you would have less phosphorylation. And so we see how on this, when you go from non-treated over to the 10 micromolar and 20 micromolar concentrations, you'll see there is this decrease in phosphorylation of lamin. So this was all just to validate that RO did work on the axis as it was supposed to. And so when I then took cells that had been treated with RO for 4 hours after they had been exposed to TGF-beta, I saw that I was able to increase the stiffnesses back to the levels where they were had they not been treated at all with TGF-beta. And so after stiffening the cells, I then wanted to see, does that finally lead to that reduction in acquired cell speed? And so once again, I've included a video here for you to see, and you notice that these cells here are much less migratory than those previously seen TGF-beta treated cells. They really just oscillate in place rather than trying to actually traverse through the channels. And we see that in both cell lines, the RO treated cells were the slowest amongst the 3 treatment groups here. As I said earlier, U87 is a little more finicky working through it, but even with the TGF-beta trends being a little more nebulous, the RO has remained consistent. It is always the slowest from there. And so to summarize the results of AIM-1, I took GBM human cells and I was able to induce these mesenchymal transitions in them, simulating disease progression with TGF-beta. When I analyzed these cells for their protein profiles, I found there were numerous alterations, including just overall lowered expression, which corresponded with decreased stiffness and increased invasion potential through confined environments. In trying to reverse the behavior using RO3306 after being exposed to TGF-beta, I showed I could reverse all the previously seen behavior, indicating that this could be an interesting axis to develop new novel therapies for treating GBM. And so looking ahead, I could look at other pathways, other mechanisms. I had focused a little bit more on the nuclear region. The cytoskeleton is still a potential door that I could go through. And of course, this was all done in vitro. I'd love to explore this in 3D and eventually in vivo. And so the next part is getting into more of the immunosuppression and immunology focused work that I've been working on. And so in specific AIM-2, I'm really focused on seeing how does ECM and stiffness contribute to the immunocapacity of the tumor cells. And so my main 2 goals are to define the role of HA-CD44 mechanosignaling and potential implications with PMT that I studied in AIM-1, as well as look at eventual immunosuppressive cytokine production. And I've included down here just the general flow of how I expect this experiment to go, where you have stiffer HA and CD44 bindings of substrate and cells, increasing in these PMT effectors that I talked about in AIM-1 to eventually get these interleukin secretions, which are known to be immunosuppressive. And so for those not familiar with HA and CD44, I'm referring to hyaluronic acid. This is going to be the primary ECM component in your brain, and GBM upregulates receptor CD44, which is what binds to HA. So this is very advantageous for the GBM to bind there. And I've included just a general schematic to show some of the different pathways that are upregulated and involved in this binding activity. So we know that HA does have some kind of pro-tumorigenic effect, and that makes it a very powerful analytical tool for looking at these models in vitro. As we know, the brain is not a piece of plastic, and so by incorporating this ECM component, you're now getting a more biologically mimetic type of culture system. And so this figure here just demonstrates some previous experiments that we've done demonstrating the importance of HA, where you can see how by varying weight percent, you can increase the stiffness to different levels. By using matrigel versus this integrin binding, you can tease out differences in invasion, 3D invasion of these 2 tumor lines. This one shows just on the different stiffnesses how you can see the invasion profiles differ there as well. And so, as I mentioned, GBM is very immunosuppressive, and that's because it has a wide number of mediators surrounding it that promote this immunosuppression. Mesenchymal GBM is classically going to be one of your most immunosuppressive types of cancers. These papers here go into detail about some of the different mechanisms and just different blood proteins and things that are involved with that. But as I had mentioned earlier, HA-CD44 binding, there is some pro-tumorigenic effect, and we see actually that STAT3 and ZEB1, these effectors that I studied to name one, they are downstream of CD44 binding. What's more, these two effectors, which you'll see in the bottom two figures here, do correlate with a high percentage of GBM patients, with ZEB1 being near 50% here. And then this just showing how in some different treatments, when they were looking at STAT3, you saw in the normal cells there was a high level of that in there with interleukin production. And so I'm looking now to try and bridge all of this together and show this direct pathway that leads from this binding to this immunosuppressive cytokine production here. And my hypothesis was that the stiffer gels, the ones more reminiscent of a tumor environment, those that are stiffer than the surrounding brain matrix, which ranges anywhere from 100 pascals to 1000 pascals, these would now be key to inducing this upregulation of CD44 binding and thus more production of these interleukins downstream. My main focus has been on generating the platforms, as these are not still well described in the literature, but eventually I would go back to repeating many of the same biological assays that I used in part one, and eventually generating some knockouts to interrogate these various points on the axis, so ZEB1, STAT, CD44, etc. And so, as I said, the integration of HA is still rather new. We see it was only really first implemented widestream back by the Kumar lab at Berkeley about 15 years ago. And in their paper that they published on creating these platforms, they had actually used rheology versus AFM. Both can give you information about the material, but rheology is used to measure shear modulus, which is more important with liquids. Whereas I'm using AFM, which is going to tell me the Young's modulus. So give me some more information about the solid type properties of the cells. In their work, they showed that by varying these thiol repeating units, so the cross-linking units that they need, they were able to increase the stiffness on these gels across various weight percents and eventually get a decrease on there. I chose to use this 3% weight line to guide me, as this encompasses most of the known biological range of brain stiffness. So in creating my gels, I first wanted to show, can I create a gel that is mechanically tunable, but does not possess these ECM signaling capacity? And so I used polyacrylamide. These are well established, used all the time. On the x-axis, you'll see the expected modulus of each gel, and on the y-axis, my measured modulus. And you'll see that for all the gels, they did line up very much with these different regions of interest that I had chosen. HA gels, though, have proven to be a little bit trickier. As I said, I started with the 3% HA line, just taking these large steps in thiol ratios. And what you'll notice is that I do recreate the increase in modulus and eventual decrease, but my ranges are well outside the 500, 1500 ranges that I wanted to look at earlier. And so I decided to lower my weight percent, vary my thiols. I actually did do a 0.25 thiol in part C, but it failed to gel up multiple times. Just it didn't have enough to create the solid structure. But what you'll see is that in these two thiol ratios, they are both lower than the previous 0.25 thiol and are more in that targeted range that I was wanting. So from these two middle steps, I began working outwards to see what was my maximum limit and what was my lower limit. And I showed that at 0.3 thiol for this 2%, I was able to get it a little under 500 pascals. And once you get up to 1 and above, the modulus doesn't change very much. So you'll see that in these higher thiol ratios, they were able to get up to around 1500. So more in that healthy, kind of soft range, and that more diseased, stiffer 1 to 2 kilopascal range. And so current findings are just that the PA gels have been consistent, not given very much trouble, but the HA gels, I'm beginning to understand a little bit more clearly and getting into those predetermined moduli values. Of course, I still have a lot to do on specific aim 2, but now that I've finished the optimization, I expect this to increase pretty quickly and complete the other goals that I had set forward, with the expected findings, of course, being in the positive. I hope I'm right, but we'll see. So the final part I won't talk about too much, as it is still quite a ways down the line, but this is just finishing up part of AIM2 and moving it into more of a 3D space. So I've included the previous line of thinking that I had for AIM2, but as I said, I'm building off of it and showing now the direct response of the immune cells and going into that 3D environment. So we know that immune cells, while often are responsible for eliminating harmful agents in the body, they're a little bit of a double-edged sword when it comes to cancer. They can be classified as either pro-tumor or anti-tumor. And this figure shows just some of the more well-known tumor cells, or sorry, the immune cells that you'll see. But what's interesting is there's an amount of cross-regulation that occurs. And one of interest are these MDSCs, specifically monocytic myeloid-derived suppressor cells. These work to suppress T cell activity and general proliferation and just binding affinity, which then reduces the T cell's ability to eliminate the tumor cells. And then I've included just a general figure to elaborate on that a little bit more. But these cells were chosen in particular, one, because we have an immunology partner who specializes in using them, but two, because they do have an inherent relationship with GBM. The brain is what we call an immunoprivileged site. Whenever someone has an immune response, the primary reaction is you're going to get inflammation, swelling, increased blood flow, pain, all these things you don't want to happen in your brain. And so the brain tries to limit the immune cells that come through. However, myeloid-derived suppressor cells are some that are permitted to go through, because in addition to their pro-tumor effect, they also just have some generally beneficial effects in the brain environment. And what you'll see is that in what's classified as a healthy brain sample versus a GBM patient sample, you can see there is a large increase in the percent of the population that are these suppressor type cells. And so in knowing that these suppressor cells are here, I wanted to tie it back to my previous aims and see, is there any relation with this PMT process that I've shown I can induce? Is there any correlation with these stiffness profiles that I'm trying to characterize and induce these differential responses in my cells? And so there's some literature to suggest, and from that, with my results that I've seen so far, I came up with this hypothesis that there is, via this axis that I proposed in specific AIM2, this way for MDSCs to be recruited now to the GBM microenvironment and essentially create a bubble of safety, protecting them from these harmful CD8 cells. And so the novel experiments are really going to focus on, again, taking this into that 3D space, using xenografts and eventually using mass cytometry. This is going to be a more advanced form of your classic fluorescent imaging that you'll see with antibodies, just giving you a much higher degree of specificity and allowing you to view many more populations of cells, as well as overlap them and spatially map these immune cells with the ECM, with the differential GBM cells, and everything else within the tumor. So it's going to be very exciting when we get there. Expected findings, of course, in the positive. And then that ends my talk in focus of my dissertation. But I wanted to still talk about some of the other aspects of work that I've been doing over the last year. Because as we know, when you're working in your PhD lab or your master's lab, even as an undergraduate, sometimes your PI just says, I've got a new project and I want you to start working on it. I don't have anybody else to do it. So you just kind of get told to do some stuff. So I wanted to talk about some of the ones that I got the opportunity to work on in addition to my primary work. And so I've just listed out the general titles here. Most of these are interdepartmental. I work a lot with Dr. Freebos. I do some work with an immunology partner, Dr. Eden Deputy, over at the other campus. And then this study here on Drosophila, probably the most removed from my basic work that I do, was done with a cell biologist in SRB. And then I do a lot of work with AFM. That's what you're going to see on here. can't really escape it. So the first project I'm going to talk about, though, I did in conjunction with Dr. Freebos, and it's taking these human GBM tissue samples that we have, and I probe them for stiffness using AFM. Dr. Freebos looks at them and their metabolite profiles, and he does his machine learning, and he's showing all this demographic data. Kind of linking these different aspects of the tumor system together. And so, as I said, my contribution is using AFM. Once again, I took these human samples, I probed them in their core, their rims, and you'll see that for the most part, they all fall within the expected ranges for brain tissue, getting into some more diseased-like states here. And what was interesting and a little bit unexpected was that for most of these, you'll notice that the rims are stiffer than the cores. And so we had initially believed that within the core of the tumor, you're going to get all these forces as they're expanding outwards, they're putting greater and greater pressure on those inside cells. We expected that to be a stiffer region, whereas the softer cells on the outside are the ones needed to bypass throughout the rest of the brain environment. But this proved to not be the case, and so it's kind of giving us some questions now. Of course, Dr. Freebos is looking at other aspects of this, using his machine learning, looking at metabolites. I don't currently have any demographic data on this, so my interpretation and speculations of this are a bit limited, but we are nearing publication on it, so I'm excited to see that and to kind of get the full picture on that, so to speak. The next project is going to look very similar to the previous one, but this was done with our immunology partner as well, and focuses on looking at these tissue samples that had been treated with a neoadjuvant chemoimmunotherapy. So all that is a fancy way to say is we applied an immunotherapy in addition to some of the other therapies on there and wanted to see what happened with them. Once again, you'll see profiles are generally within the ranges that we would expect to see. Once again, you'll see that in most of the patients, the rim ends up being quite a bit stiffer than the core. Again, without demographic data, it's a little bit more difficult to speculate on what these findings could implicate, but these projects are still ongoing and there is still a lot of room to work and to further analyze what is happening. So the next part is going to get into this fly study, and let me just real quick. Just need to stop the recording. Did it stop recording?
Not yet. I'm trying to